Welcome to Life in Biology. I'm Dr. Joel Graff. This is episode number 12, and today we're going to be talking about big picture applications for CRISPR. I got a little tongue-tied at the end of my last video. I tried to talk about applications in the last 10 seconds. It really is going to take maybe a couple videos. So just to recap and to let you know the plan, to recap, I taught you in the previous video that Cas9 is a protein that you need along with a guide RNA, a single guide RNA, in order to create double-stranded breaks in the DNA at specific places in your genome. Now the cool thing about this CRISPR system is that with this guide RNA, it's programmable. So you can swap out some of the sequence in your guide RNA, and you can make the CRISPR system go and cause a double-stranded break in one spot or another spot very easily. So as far as applications go, this is the first video, and I'm just going to discuss some big picture applications. The next video I'm going to get into a little bit of the nerdy nuances or variations on the theme of how the CRISPR system can be modified to do other tricks. So I want to take a second to talk about genome editing in mice. So CRISPR is good for doing genome editing, but we've been able to do this since the late 1980s. Okay, so in the late 1980s, some scientists, Kopecky, Evans, and Smithies, they were awarded the 2007 Nobel Prize for their work where they were able to do gene therapy on a mouse. They had a mutation in a gene called HPRT that they wanted to fix, and they were able to show that they could fix it in one mouse and that could be passed on to the offspring of those mice, and so you can over time fix the mutation. And really today, transgenic mice are the workhorse of many areas of biology, especially I'm focused on immunology, and you see transgenic mouse work all the time. There are some drawbacks to doing genome editing with the drawbacks with the current technology or the pre-CRISPR technology. There's one is it's expensive. So I often heard the number $100,000 thrown around when you talk about making a new mutation in mice. It's labor intensive. There's a lot of DNA constructs to make. Then once you did have your constructs and you did get it into a mouse germline, once you have your mouse, then you have to do all sorts of what they call back crossing, and so there's like 10 to 12 to 15 rounds of breeding that you have to do, and not only is that labor intensive, but it takes a long time to do 15 generations of mice. Another problem is that mice aren't humans. Mice have been great. We've learned a ton from them, and we're still learning a ton, so it's not time to shut down the mouse industry yet, I don't think. But mice aren't humans, and it's becoming clear that in certain situations, what you learn from a mouse doesn't always translate well to a human. Some situations it does, some situations it doesn't. So that's a drawback. And then it's not easily multiplexed. By multiplex is, what if I want a mouse that has 1, 2, 3, 4 different gene changes in it? Well, I could, what I would do is I'd get strain 1 that has the first mutation, strain 2 that has the second mutation, strain 3, strain 4, and then I'd have to do a very labor-intensive breeding program to cross these against each other until you come out with the mice that have all the different changes that you wanted. So that was also very labor intensive. So now with CRISPR, it's the solution to a lot of the problems we have for creating transgenic mice. Now, just because I keep saying mice, the thing is now that it's so easy to do CRISPR to do genome editing, you can do it in pretty much any organism you can think of. I use CRISPR in a parasite, a single cell parasite called Leishmania. You can do CRISPR in bacteria, you can do CRISPR in plants, you can do CRISPR in humans and other animals. And the as far as if you're talking about genome editing in mice, it's basically a thousand-fold reduction in cost. So with CRISPR, you get 99.9% discount. And the reason for that is that being able to create a double-stranded break exactly where you want it allows you to repair that double-stranded break with the DNA sequence of your choosing. So George Church is a fairly famous scientist, and he's also very good about communicating with the public. I'm linking to I think it's a Huffington Post article below, where they were interviewing George Church, and he gave his top 3 ideas of what he's excited about with CRISPR. And so I just thought I'd take a few minutes to talk about this. So the first thing he's interested in is gene drives. And so gene drives, this is probably going to be a complete separate video to completely explain this, but the idea is that if you want to change the genetics of an entire population, you couldn't do it with normal breeding, because if you released, let's get mosquitoes for example. Mosquitoes, we could cause a mutation that would make them resistant to the parasite that causes malaria, and if we could get the whole population to have that mutation, then you can wipe malaria off the face of the earth. So that's a pretty good goal, huh? Well, the problem is that if you release some mosquitoes that do have resistance to malaria into the wild, it's just going to get diluted out into the wild population and not take over. But gene drives are a new technology that allows it so that every offspring that comes from a mutant organism will have mutant offsprings, or I shouldn't say mutant because that has negative connotations. Every offspring from an organism that has the desired feature, all the offspring will have that desired feature. But we'll talk about that in another episode, ethics and also safety involved with that. So you can also improve crops. We've been using transgenic crops for a long time. Most days you probably eat some sort of genetically modified crop, and so you can now, with this technology, create more unique and diverse and complex versions of transgenic crops, such that you can introduce an entire metabolic pathway that'll make a vitamin, for example. So you can have potatoes that have high level of vitamins in them. Transgenic pigs. So why would we want to create transgenic pigs? Well, pigs are about the same size as humans, and so organs from pigs could be used for transplant. And transgenic pigs, I believe George Church probably mentioned this one because there's an example out there. I believe they've created a pig with 68 different mutations in it using the CRISPR system, and that would pretty much be unthinkable prior to the CRISPR system because it would just take too much time, too many generations, too much effort. But with CRISPR you can make multiple changes to a genome in a single go, and now it's just coming up with all the things you might want to do with CRISPR. So hopefully that helps understand some applications of CRISPR, and we'll get a little bit more nerdy next time. Thanks for watching.